All right, good afternoon. We are getting sparse. I know everybody's busy, has a lot to do. Hope your lab exams are going well. So you probably had those already this morning. Let's jump right in. I'm not going to review too much with you today, but whatever time we have left on Wednesday, we'll review. I anticipate we will have some time there. Just remember there's not a lot of homework, but make sure you check through. It's kind of scattered here and there among those last 5 or so chapters of OpenStax. I did want to just drop back just a minute to reiterate or maybe review a second about our smallpox here. So smallpox, oops, button there. Smallpox family Poxviridae, that's kind of intuitive. The genus name Orthopoxvirus, maybe not as intuitive, but it does have that pox root in there. We said that smallpox is caused by the variola species. There's 4 really closely related species here. The variola, the vaccinia, that's the species that we used for the original smallpox vaccine. And then, of course, cowpox was the species that Edward Jenner used for his first, or what we attribute to the first vaccination. So he took those sores from somebody that had cowpox and then kind of pressed them into other people's skin. That's kind of what we're replicating when we did the original smallpox vaccine. So this needle, I read a little bit more about this. That original smallpox vaccine, they didn't call it a shot. They basically, this is a special kind of needle to just poke the skin. So little tiny stabs, kind of like the way Edward Jenner would have done it in the late 1700s. So, you know, little stab enough to really make cause some bleeding, a little bit of a sore that took a couple of weeks to heal up. And then, of course, monkeypox or mpox. So some of, we'll get to today, some of the reasons that monkeypox is emerging, we think, is because people are no longer immune to smallpox. It's been long enough since anybody's been vaccinated. Some of the same drugs that we would use for smallpox if it emerged are being used for monkeypox today. All right, so double-stranded DNA virus, just like all the herpes viruses. More about monkeypox. So this is an infection that was first recognized in about 1940. And from time to time would have outbreaks outside of Africa, but only kind of a few cases. So this is the first time that it's really traveled. The WHO, the World Health Organization, doesn't consider it epidemic or pandemic. It calls it a public health emergency of international concern. Okay, so apparently hasn't gotten to enough cases where they would consider it epidemic or pandemic status. In the US, just some numbers, there's been about 29,000 cases and only 15 deaths in the US. So when we look at the case fatality rates, the case fatality rate is typically considered like 3 to 6%. We're not getting to that in the US. Apparently, you know, good enough treatment for really complicated cases that we haven't got to that. Internationally, about 75,000 cases, according to the WHO. Okay, so again, traditionally, people would get this when they came in contact with some animal reservoir. There's a number of non-human primates, monkeys, that harbor this disease, but other things like the Gambian pouched rat or tree squirrels, rope squirrels, certain species of mice. So not just monkeys, but some other small mammals. So traditionally you get this by contact with these animals. Interestingly enough, with this outbreak, about 96% of people that become ill have been men. I don't know if there's anything special about that. Or again, like we saw the estrogen possible connection with the Epstein-Barr virus and the Burkitt lymphoma. I don't know if I haven't heard why that is. Many of the cases in the US have been men having sex with men. That's how it's been transmitted, but there's nothing special that makes this a sexually transmitted disease per se. So again, traditionally it's been zoonotic. This is really the first time that we're getting person-to-person transmission. And again, what we think is contributing to that, first cases of that person-to-person transmission was in 1970. So again, people are not as commonly, anybody under 50 has not been inoculated or vaccinated against smallpox. So older people maybe have a little protection against this, certainly against severe disease, but not younger people. What do the symptoms look like? And I've talked to a couple of people that have seen cases in Grand Rapids that work in the hospital here. Now we're down to about 1 case a day in all of Michigan. So the outbreak is certainly waning, at least in Michigan. It is still spiking in some other states in the US. What do the symptoms look like? Fever, intense headache, swollen lymph nodes. And then maybe 1 to 3 days later, the rash breaks out. And it's kind of a peculiar, a lot of rashes will start out in the areas of your body that's like the warmest, like your trunk region. That's what we talked about for chickenpox. This you get a rash, palms of hands, soles of feet, which that kind of reminds me of hand, foot, and mouth. I've seen that before, but other places too, oral mucous membranes, less commonly genitalia in the eyes. Okay, so usually self-limiting, lasts 2 to 4 weeks. Pretty painful though, like not something that you would enjoy experiencing. Again, typically 3 to 6% mortality. Again, we are not seeing that in the US, but perhaps more worldwide when access to healthcare isn't as good. Okay, and cases can be more severe in children when they get it, and there's lots of possible complications, encephalitis, inflammation of the brain, loss of vision sometimes if it gets in the eyes. Okay. Transmission. So it has to be really prolonged face-to-face contact by respiratory droplets. Okay, this is not passed nearly as efficiently as smallpox. You know, even with some prolonged contact, you know, a pretty low percent of the time does it get passed. Or close contact with infected lesions or bodily fluids. So the sexual transmission, perhaps likely, but I don't know if that question has been answered for sure. Rarely by fomites. So maybe exposure to sheets that an infected individual has slept on. Okay. Let's see. So again, majority in men for some reason. The R naught here is about 1.29. So that's considered low. So an infected individual probably would give the infection to maybe one other person that they come in contact with. So that's considered fairly low. Just because cases are waning in the US, that doesn't mean that we're not worried about it anymore. So health officials still think that it has the potential to not be controlled well. So it could mutate, it could change a little bit, and suddenly it would kind of become more of a threat. So, all right. What do we have to treat it? Well, there's this drug, tecovirimat, that was developed in 2019 for the variola virus, for smallpox. Okay, for variola. Okay, so again, smallpox disease. So interestingly enough, it's been used, it's not approved by the FDA to be used for monkeypox, but, sorry, mpox, but it has been approved by compassionate use, I guess, sort of an emergency way. So I think it's very interesting that suddenly we have this new drug for smallpox in 2019, 40 years after the last smallpox case has ever been. So that tells you how worried the government is that some bioterrorists are going to suddenly unleash some smallpox. There's supposed to be only 2 vials of smallpox virus frozen down in a freezer. One is in the CDC in Atlanta, and one is in a lab in Siberia at a viral institute in Russia. So there's supposed to be only 2 vials of that, but still the government has bought up 2 million doses of this tecovirimat just in case there'd be some kind of biological warfare. This also goes by the name T-pox with 2 X's. I don't know if that's it's probably some kind of brand name for tecovirimat. Okay. So how does this work? It inhibits the virus from leaving the infected cell. Okay, so it actually inhibits one of the major envelope proteins on the surface of the cell. Okay. All right, so we don't have a lot of data on that when it's used for monkeypox, but it does seem to lessen the chance that somebody will die from the infection. All right, so the vaccine that they're giving people for mpox is called JYNNEOS. So JYNNEOS, I'm not sure what that's an abbreviation for or what that refers to, but that is FDA licensed for smallpox and monkeypox, sorry, mpox. It's a live attenuated virus. So it's another kind of orthopox virus, but it's one that is non-replicating. Okay, so it's been weakened so that it can't replicate. 2 doses, and that was approved in 2019. So that vaccine that people are getting for mpox is not the same one that we used 50 years ago for smallpox. You don't have to have all the little pokes. Okay, there's another vaccine that is available, but it has a few more adverse effects. So I thought we wouldn't mention that. Okay, any other questions, comments about mpox, smallpox? Again, the threat of smallpox really low, but the worry is that you know, somebody will get a hold of it. Yes, question. So good question. If we're worried about some bioterrorists, why aren't people vaccinated? Originally, it was because that original smallpox vaccine just was so nasty and made people so sick after it. If we had a better vaccine, then maybe that will be the case. As hard as it has been to get people vaccinated against COVID, I would guess that most people would say no thanks. But it's interesting. At this time, it's not available just for anybody that wants it. But Yeah, that would be a good discussion for some medical ethics class. Yeah. Okay. All right, let's move on to some RNA viruses. There's no other questions. Okay. All right, so we've got 3 RNA viruses. There are many more interesting ones, and I sometimes will pick different ones, but we'll stick with flu, HIV, and coronavirus, I think this semester, and then hopefully have some time left. Okay, so we'll start with the orthomyxoviridae. This is a large family of viruses that primarily cause influenza. And just to kind of set the record straight, it's for me, it's like nails on a chalkboard when somebody says, oh, I had the flu, I was throwing up all weekend. Influenza is a respiratory infection. So people rarely vomit when they have a respiratory infection. So, sometimes kids do a little bit more often, but adults will rarely throw up. So when somebody says they have the flu, they should at least say stomach flu if they've been throwing up. And even that is kind of a misname. It should be like enterocolitis or something like that. But when we say influenza, when we say flu, I'm talking about a respiratory disease. Okay. There's 3 well-known types of influenza in humans. We refer to them as influenza A, influenza B, and influenza C. Influenza A is typically the most serious in terms of symptoms, most serious disease. That's the only kind that will also infect animals as well as humans. Okay, so we'll get back to that in a minute. Influenza B is usually a little bit less serious, and the ones that cause, the epidemics and the pandemics are usually not B, they're usually type A. And influenza C causes a rather mild disease, almost fairly benign. Okay, so what makes the A ones more serious? Usually they are changing much more quickly, so people don't have as much immune response to them. The B ones change a lot more slowly, apparently. Okay, so what else about these viruses? They're enveloped, and they have a helical capsid. Remember, the capsids are either helical, polyhedral, or complex. So it's kind of hard to see that here, but you can kind of see it in that picture a little bit. They have a segmented genome. And influenza A usually has 8 segments, and influenza B usually has 7. So you can see here, this must be influenza A variant, 1, 2, 3, 4, 5, 6, 7, 8 segments. So why does it have segments? Of course, an unsegmented genome would just have all its nucleic acid in one long piece. RNA viruses especially tend to mutate quite a bit. So I guess if the genome is in segments, if you get a mutation at the beginning, and then if it was in one big segment and you had a mutation at the beginning, maybe none of the genes would get transcribed or replicated correctly. So if it's in segments and you have a mistake in one, maybe the other segments will still make a viable variant. So high mutation rate. What kind of genome is it? It's a negative sense single-stranded RNA variant. So this variant would have to bring in some RNA-dependent RNA polymerase. So by RNA-dependent RNA polymerase, it would use that negative sense single-stranded RNA as a template to make positive stranded single-sense RNA to be read at the ribosome, translated by the host into proteins. Okay, so it's got to have a copy of that enzyme. There are spikes for the influenza virus. We're going to spend a little time talking about those glycoprotein spikes. There's two types, hemagglutinin, usually abbreviate that H. Those are the big red ones here on this drawing. Those are necessary for the virion to attach to the host cell and also fuse with the host cell. So that's hemagglutinin, and then the smaller green spike that you see there, that's neuraminidase. So we'll abbreviate that N. And that's necessary for release of the new virion from the host cell. Okay, so there is about 16 different types of hemagglutinin in the influenza A viruses and 9 different types of neuraminidase. Okay, so this is one of the ways that, but not all of those types infect humans. Actually, I said that wrong. So 16 different H's, 9 different N's. This is one of the ways that we name strains of influenza. So just as an example, it looks rather complicated, the strain names, but if you break it down, then it starts to make sense. And we'll look at some of the strains that are included in this year's flu vaccine. So here's an example of strain nomenclature. So this first one, that's whether it's influenza A, B, or C. Okay, so to influenza A, the next thing here is the animal of origin. Okay, and if there's no animal listed there, then the strain was first found in humans. Okay, so equine, horse, swine, pig. Where it was first isolated, that could be a city, it could be a state, could be a country sometimes. Okay, and then this number is just a strain number, the year in which it was first isolated, and then the combination of, or what type of hemagglutinin and what type of neuraminidase. So a lot of people, you might have heard of H1N1, swine flu. That's what the H's and the N's stand for. Okay, so the spikes, that's the most antigenic part of the virion. That's what our immune system notices the best. So that's what we use for the vaccine. Now, there's a couple of different ways. I said that flu chain influenza viruses, they mutate a lot and they change pretty quickly. So there's a couple of different mechanisms that the variability of those glycoprotein spikes will change. Okay, so we talked about antigenic variation with Neisseria gonorrhoeae also. It's a little bit of different kind of antigenic variation, 2 different kinds that we see with influenza. And with influenza, it happens really slow over a period of months and years. With Neisseria, remember, that could happen, you know, in a period of like 7 to 10 days, where those fimbriae or pili change their protein composition. Okay, with influenza antigenic variation, it's much more slow. Antigenic drift is a kind of antigenic variation where we just get some kind of slow point mutations build up over time, slowly over time, gradually, to kind of subtly change the composition of those spikes. So because of that, we think that causes kind of a biennial difference in flu. We track flu cases really closely worldwide. And usually, on average, we have 1 year where flu's pretty bad, and then the next year it's not so bad. And then again, because of these subtle changes, we have a worse year. The next year, people still have maybe some response to those strains that were circulating. So we kind of have up and down response. And again, these are gradual changes because that RNA dependent RNA polymerase makes a lot of mistakes. Okay, so biennial outbreaks due to subtle changes over time. Antigenic shift is a much more abrupt change in those glycoprotein spikes. And only type A influenza does that because that's the only type that also infects animals. Okay, it can cross species. So antigenic shift is when two or more viruses from different, usually from different organisms, infect another organism, and you get reassortment in those new variants that are produced. So that's what this graphic is supposed to try to explain that a little bit better. Here with antigenic shift, say you have a virus A, let's say it's H1N1, and maybe this is in a pig. And then you have another virus, let's say H3N8. Maybe this is in a duck, for example. And maybe those are both coming together in a third organism. And then you get reassortment of those two different viruses in a third organism, and you come out with some virus that's totally novel. So maybe an H1 from virus A and an N8 from virus B. Okay, so you end up with something totally different that people haven't seen before, that wouldn't have much of an immune response to. And we think that this is going to be responsible for occasional outbreaks of really severe illness. Okay, so the last time that we had severe flu was about 8 years ago. Well, it's been more than that. Sorry, my math. 2008 is what I meant to say. That year, 2008, 2009. This doesn't have years on it. This doesn't correspond to anything. On average, we have bad outbreaks about every 10 years. So we've been saying for a few years, oh, we're due for bad flu, we're due for bad flu. And that's why you hear a lot of, hey, get your flu shot, get your flu shot. We're just kind of due for some reassortment like that to happen. Okay, so here, antigenic shift, remember, or sorry, antigenic drift is just more subtle kind of over time. Okay. Transmission of influenza A, that's probably, you can probably guess at that, large particle droplets. So most commonly, sneezing, coughing, close contact, within 1 meter. It has the potential to be airborne, but not over great distances. Some fomite transmission on some inanimate object, it probably would be viable for about 1 day on a doorknob or a computer keyboard, something like that, which is kind of a long time for a membrane-bound virus. Okay. Let's see. Of course, it's going to be more easily passed in areas with high population density. So, a jail or an army barrack, for example. So that leads me, I want to at least mention that flu pandemic that happened about 100 years ago. It's very interesting. There's a really interesting book if you're interested. There's a book by Barry. It's called The Great Influenza. If you are looking for a good book over the holidays and you're interested in reading about this sort of thing, that pandemic was not like the normal flu that we get from year to year. Flu's bad, and we're going to talk about that in just a second. But this particular strain caused some unusual symptoms. People would bleed often from the nose. There was neurological complications in a number of cases. In fact, we think that Woodrow Wilson, when he recovered from the flu, he made a lot of decisions that were totally different from the way he had acted before he had the flu. So we think that that affected him that way. It had a very high mortality rate, about 5% mortality. It's estimated that a third to half of the world's population was infected. It came at a bad time. People were moving all over the world due to World War I. And living in close proximity to each other, at least people in the military. It's estimated that 50 million people died. And normally the flu hits people that are very young and very old much worse, but this strain, it was people in their prime between the ages of 18 and 40 that had the highest mortality. So we think that that was due to their beefed up immune response that caused a cytokine storm. Okay, giving away a homework answer there if you haven't done this homework yet. But you know, an inappropriate immune response. It's a difficult thing to, I don't know whether they had drugs 100 years ago that dampened the immune response like steroids, but it's kind of a difficult thing because you still need your immune system to fight the virus. It's just, again, too much cytokine release can be a bad thing. So we haven't seen anything like that. The number of people that had pneumonia as a result of that was pretty high. Of course, this was before they had antibiotics. And you could argue that we would be a little bit better suited to deal with bacterial pneumonia as a secondary infection today. It's what they were then. They're actually pretty good. They could immunize animals like horses and harvest antibodies and use those antibodies to treat people with bacterial pneumonia. So they were pretty good at that, not when they were overrun like this. This is a school gymnasium. They had to set up cots. Imagine the level of care that you would get, something like that. It just sounds terrible. The number of doctors and nurses that died was astonishing. So interesting to read about. Are we better situated today to deal with something like this? Somewhat, but we still don't have good drugs to treat flu. So we'll talk about that in a second here. Clinical disease, typically for influenza, about a 2-day incubation. So a couple days after you're exposed to somebody, those virions bind to the respiratory tract and cause a lot of inflammation and cell death. Let's see what I'm going to say about that. So you know the symptoms. You probably had this at some point. High fever, chills, body aches, bad body aches. Well, that's a different story. I'll tell that later. Lethargy and cough. Again, vomiting sometimes with kids, but not usually with adults. An ordeal, 7 to 10 days. Kind of goes on and on and on. Okay. And again, secondary infections like ear infections, pneumonia, particularly dangerous for elderly people and really young children. Okay, so what do we have to treat this? We have a couple different kinds of drugs. There's actually 3 different drugs that are considered neuraminidase inhibitors, and they abbreviate that NAIs, neuraminidase inhibitors. There's actually 3 different ones. We'll just mention oseltamivir. The trade name of that is Tamiflu. So I'll just call it Tamiflu because that's the most recognizable thing. We've had this since the late 1990s, I believe. And again, this is going to inhibit the release of virions from the host cell. Okay, again, there are a few others besides Tamiflu, but that's kind of the one that's most popular. So it always helps a graphic to see this. So if you look at the neuraminidase activity, Here's the new virion that's leaving the host cell, and you can see the hemagglutinin spikes are still kind of attached to these sialic acid receptors that are on our cells. And then the neuraminidase, it kind of cleaves that, and so the new virion will release. In the presence of these neuraminidase inhibitors, that just keeps the neuraminidase from cleaving that connection and if it stays stuck to the host cell, then it's not going to go off and infect another cell. And if it does detach, then the new virions tend to really stick together quite a bit, and so they're going to be more easily noticed by the immune system. Okay. The other drug that's newer, was just approved about 4 years ago, is called baloxavir. The trade name for that is Xofluza. Okay, so that's the one that you see on the commercials, you know, flu really sucks the life out of you or something like that. Okay, so this is a newer one. This is a different mechanism. It's not a neuraminidase inhibitor. It inhibits cap snatching. Okay, so I couldn't find a good picture for this, so I'm gonna have to draw it. If you remember back when we talked about genetics, eukaryotic transcripts of messenger RNA. We said that those RNA, they have a little bit different, more processing than the prokaryotes have. They have a on the 5' end, they have a guanine cap. Okay, and on the 3' end, they have some polyadenylation. And we said this is necessary because that transcript has to travel from the nucleus out into cytoplasm, where that 5' cap is what lines up at the ribosome to get translated. Again, to spell ribosome. Okay. Okay, and there's introns and exons in there. The intervening sequences get cut out, and the exons, the express genes, are what stays. Okay, so what the influenza virus does, it has endonucleases. Let me just change color here a second. That will cut off this 5' cap, and then attach it to the viral RNA. So now it can much more easily be read at the ribosome. Okay, so that's cap snatching. And so what this drug does is inhibit that cap snatching. So now the influenza RNA, it just has a much harder time getting its proteins translated. Okay, so if we compare these two drugs, Tamiflu and Xofluza, let's do that. So both of them, same. You have to give them to the patient as early as possible in the infection, ideally within 48 hours. Usually if you give them later than that, they don't do any good. You've got to try to inhibit that viral replication early on. After a couple days, you know, it's the host responses, the inflammation that's causing a lot of our symptoms and causing us to feel lousy. So for both of them, you got to give them right away. And unfortunately, they are both by prescription only. So you're feeling lousy and you got to drag yourself to the doctor. There's talk of making Tamiflu over the counter, but I checked and that is not available yet. So that would make it maybe a little bit easier to take that. Let's see. Costs is the next. Oh no, how often do we give it? Tamiflu, you give 2 doses a day for 5 days. doesn't seem too bad. It's a pill, orally. Xofluza, it's only one pill. Okay, so you only have to take it once. Cost. Tamiflu is generic now. It's been around for 20 years or so. So the last I looked, $20 to $50 somewhere for a dose of that. So not cheap, for sure, but it was over 100 before it was generic. Xofluza, a little bit more money. Okay, so if you're paying out of pocket, that might play into the equation whether to take it or not. $150 to $180 for one pill. Again, this just came out in 2018, so it's still pretty new. Who can take it? Tamiflu, they'll give it to babies, 2 weeks and older. I fact checked Xofluza just like an hour ago, and now we are giving it to ages 5 and up. Okay, so when it first came out, they hadn't maybe tested it properly in kids. Now they'll give it at least to ages 5 and up. Side effects for Tamiflu: dizziness, nausea, vomiting, diarrhea. A lot of potential side effects. Not for everybody, of course, but the side effects could be worse than the flu almost. Xofluza has some of the same side effects, but early reports or the reports that I have read, a little bit less chance of side effects. And of course, any drug you take, there's a percentage of people that are going to be allergic to it. Anecdotal evidence is always, fraught with difficulty, but it's powerful. I had a friend whose 2-year-old took Tamiflu and went into started having seizures, and it took the hospital a few days to figure out that it was from the Tamiflu and not from the influenza. So, and it was just an allergy from the Tamiflu. So the question is, if you get the flu, is it worth it to take one of these drugs? For both of them, is it worth it? It will shorten your illness somewhere between 12 and 24 hours. Okay, so if you would have been sick for 7 days, now you're only sick for 6 days. Is it really worth it? It's not great. You have to weigh all these things and somehow decide right when you first get sick. Well, if you knew how serious it was going to be, maybe you would want to take it, but all these things you don't know. Doctors recommend anybody that's immunocompromised or very young or very old that they should take one of these treatments. For healthy people, anybody can take it. Doctors will prescribe it, but maybe it's not going to do you a lot of good. Okay, we have lots of Tamiflu stockpiled in the government in case we ever have a bad pandemic, but it may not do a whole lot of good. Okay, so it might be better just to try to prevent influenza. We have many different varieties of vaccines. There's vaccines that are not grown in eggs. If you have an egg allergy, they have like double boosted vaccines, much stronger vaccines for elderly people. They have attenuated vaccines that you just breathe in that works better for kids of a certain age. It's an attenuated virus that will only replicate in the colder region of the nose, so much milder disease. But most of us will get an inactivated virus that's injected. And unfortunately, we have to get it every year because strains of flu that are circulating change from year to year. So we change that formulation. It's usually a quadrivalent, 4 different strains that they include in the flu. And don't memorize that. I just put that up to show you how it's a little bit different than last year's formulation. But they decide what to put into the flu vaccine. They decided in February of this year. So that's one of the problems why the vaccine is at best only 40 to 60% effective, because we have to decide so long ago in order to grow this up in eggs. It takes a long time. So if we could be a little bit more flexible and maybe decide in July, for example, and see what's circulating in the Southern Hemisphere during their flu season, then we would have a more effective vaccine. So hopefully, eventually, we'll move that way. They almost always include this strain. It has a special name, pandemic 09-like virus. That was the strain that was in the, the last time we had worse flu, that H1N1. But again, it changes from year to year. Can you get flu from a flu vaccine? No, all right? They do quality control checks. These are dead, inactivated viruses. Now, you might be sore, you might be kind of achy, especially your arm, and you could have the misfortune to get infected with a strain at the same time that wasn't included in the vaccine. And the vaccine does take a couple of weeks until you get those memory cells built up. But you can't get the flu from the flu vaccine. So hopefully help dispel that myth if you hear anybody say that. Okay, any questions about flu? I knew that would take us a while to get through that. Okay. All right, so let's start HIV, at least kind of, anyway, get started with that. So this is another RNA virus in the family Retroviridae. Okay, all the members in Retroviridae, they're considered, that just means they can carry that enzyme reverse transcriptase that makes them kind of unique. We'll talk more about that in a second. Lentivirus is the genus. That's a little bit less familiar, but human immunodeficiency virus is the one that's still considered a pandemic. I just looked that up, and still, we're getting a lot better at treating it, but still many new cases a year. So if we look at the structure of this, once again, it's enveloped. It has a segmented genome. It actually only has 2 copies of one segment. We still consider it segmented, and it's positive sense single-stranded RNA. Okay, influenza negative sense, HIV positive sense. So it would kind of make sense for this virus. It doesn't need to do anything. It can just be read at the ribosome, but it doesn't do that. The virion contains 3 different proteins that it brings along with it. Okay, so one of those key enzymes is reverse transcriptase. Okay, and we talked about that enzyme before. So that means the virion takes its single-stranded RNA, and from that makes a negative single strand of DNA, and then it also copies that complementary strand as a template to make double-stranded DNA. And then it will use that double-stranded DNA and use the host enzymes to make RNA and follow kind of the central dogma. Okay, so why does it do that? Well, double-stranded DNA is a lot more stable, and then it can integrate into the host DNA. Okay, so the other enzymes that it will bring with it are integrase. Okay, and that's so it can integrate into the host DNA, as well as an enzyme protease that's important for maturation of the virion. So what that means is when the new virion's released from the host cell, that enzyme is really important to make some cuts in some of the proteins in that virion so that it's a viable virion. Without those cuts that are made in the new proteins that were translated, then it's not a virion that's going to be able to infect other cells. Okay. Also, a couple of key glycoproteins. This figure up here is a little bit more complicated than what I'm interested in you knowing. This GP120, that is these purple heads there. It's an envelope protein. It's important for attachment to the host cell, and GP41, that's this red portion here, so that viral envelope can fuse with the endosome that it, and basically in the uncoding process. Okay, so we'll look at kind of the path that virus takes during infection, look at some treatment that's used now, and talk a little bit about coronavirus next time, as well as hopefully do some reviewing. So we'll see you Friday for our last class.